Okay, so we are talking about quantitative proteomics using mass spectrometry, and we have discussed variety of instrument platforms, variety of approaches which one can use to do the quantitative proteomics, right. So, as you can know that during the collision induced dissociation, when collision energy is subjected at that point, these peptide bonds they are broken, and then you can see B ion and Y ion getting generated. So, Y ions are these positive charge which are retained on the C terminals of the peptide ions, like on this side, and that is why the Y ion series starts from the C terminus, and then B ions are those which are charge retained on the N terminal side. So, then you have B ion series starting from B1 to B4 from the N terminal. So, these things you have already been aware now. Question is why Y ion series is more intense than the B ions, because if you look at the actual data you will find that there are many Y ions they are red, whereas very few B ions they are actually the red, right. And the main reason for that is that basic side change, especially when you are doing the trypsinization for the cleavage which happens at the lysine arginine residues, they are generating the positive charge at the C terminus, which is the part of your Y ion series, and therefore, because of this enzyme cleavage which happens at the arginine lysine residues, you will have much more intense Y ion series as compared to the B ion, right. Okay, so we had then talked to you about mascot search, especially one of the open access database software available to do data analysis, and you can do variety of analysis over there, peptide mass fingerprinting, especially if you have data coming from the multi-top based platform, multi-top platform. Sequence query, you can do MSMS ion search. There are example files, there are description for each parameter. I think those you should read in much more detail. And as you have seen this example, that how if your entire sample preparation, in-gel digestion, everything has worked out well, and then your database search is able to pick up the right protein, then you can see a good score for a given protein, and then you can establish the identity of the protein with good confidence. So, if you look into each of these peptide, you can then start seeing the B ion and Y ion series. What is important here, as I mentioned to you, that if you zoom into one of these region, you will see almost all of the Y ions and only very few of the B ions, right. This is what is expected. Now, how to cross check this sequence information, is this sequence correct or not, or in other way, in which way can you establish the sequence assignment yourself, can you establish the sequence identity of these amino acids and peptide sequence yourself. So, as you can see here, like I have kept it hiding for the time being, if you start looking at Y ion series Y7 minus Y6 for example, that gives the difference of 57, which corresponds to amino acid glycine. Now, if you come to Y6 minus Y5, you are finding 129, which is you got another amino acid. Now, if you come back here on the V ion series, important thing here is that you are cross checking the data, okay. You are cross checking data, and if now from the B ion also you are getting the same difference, then only you can see the glycine is correct. So, it means both the side, both ways, you have done Y7 minus Y6 and you have done the B1 minus B2. So, what you have to keep in mind that you are doing the from the Y7 minus Y6, not Y6 minus Y7, not in that manner. Y7 minus Y6, which correspond to 57, correspond to glycine, and if you have cross checked that for B1 minus B2, you are getting the same difference of 57, and then you can be confident that this is going to give rise to the same amino acid, and in this manner if you derive, now you can derive this entire amino acid sequence, okay, and peptide sequence can be derived from this. Not every B ion is available to you for calculation and cross check, but as long as you can cross check 2 or 3 which are the red in color, the intense B ions, then you are very confident that these amino acid sequences are correct. So, despite that you may not be able to read all the exact differences in your table and sheet given to you, but as long as you can cross check from both the sides, C terminus, N terminus, that these differences are almost similar, then you can be confident about that amino acid being correct. So, this way one could actually manually derive these peptide sequences, and although from the software you can immediately get this information because these values are already fed there, but by now I think you can do this thing yourself. There are many factors which also generates how good these fragmentation ions can be, right. For instance, if you have the loss of water from the side chain of serine and threonine, that is going to affect the fragmentation pattern, and then your fragmentation may not be as good as you would expect. Same if you have the hydrogen sulfide released from the cysteine, that may affect the fragmentation pattern, or loss of ammonia from glutamine and aspartic acid, those are also going to affect your fragmentation pattern. Proline being unique in nature, having the cyclic structure, is always creates some sort of challenges for the analysis part. So, it is resistant to the fragmentation. So, if a proline is just before or after the lysine and arginine residues, you will see there is a problem in the cleavage, and therefore, again your fragmentation and the efficiency of cleavage will be affected. So, therefore, there are different enzymes being also used if you think your protein is rich in the proline amino acids. Okay. So, then you will, in addition to the thing about trypsin, you should use chymotrypsin, you should use other peptidases as well. So, different peptide bond they fragment differently, especially because there are certain amino acids near those there are very intense B ion being produced. So, therefore, aspartate, glutamate, they are going to create some anomaly in terms of the reporter ion intensity. So, what it means that it is not always that your collision energy and your sample preparation are the only factors. The kind of abundance of amino acids present in your given proteins is also going to govern how good your fragmentation going to happen and what kind of intense ion you are going to see that. Okay. I am just briefly covering about the quantitative data analysis, especially the iTRAC based workflow. As you are aware that you have to do the pre-fractionation strategies to simplify the complex proteome, right. One strategy which you have seen is off-gel fractionation based on liquid isoelectric focusing. So, if you have done IEF and you have collected 20 to 24 fractions, each one of those fractions you are going to run in the mass spec, right. And when you are running in the mass spec, you are going to look for how good the peptide patterns are coming, the chromatogram is coming for each one of those fractions. Because if you have just run 20 minutes run for the entire complex proteome, still you will get this kind of pattern, and if you have now run 20 fractions each around 20 to 30 minutes time, now you are seeing this kind of pattern. So, it means much more number of peptides you can now read, because the MS scan rates are fixed, you cannot change those, right. They are very fast, but still the way your ions are passing in the mass spec and the way the MS scan speed is there, these two things are you have no control. So, one way you can control that to have the pre-fractionation, so that less number of peptides and less number of ions are coming each time, and then later on you can pull all of this information to generate the entire proteome coverage, because proteome, when we say we are interested to know about, let us say, all the 5,000 or 10,000 protein in a given sample. So, in that way coverage becomes very important, and to do that people follow this strategy in the iTRAC based workflow. You can see the MS ion, like if you just pass through, you will see certain MS fragmentation pattern. If you do MS, now you can generate the reporter ions, which could be used for the quantification. While I will not talk in much detail right now about the parameters, there are various platforms available for doing data analysis for iTRAC. One platform which you will see today is based on the commercial setup what we have here available, but you can still use even Mascot if you do not have the specialized software available. You can still use Mascot for doing data analysis. Only thing is, if you have access to the Mascot complete database, probably your search will be much more refined. Nevertheless, in this case, one need to define the range, one need to look at the scan time range, one need to look at the retention time, M by Z tolerance, what is the precursor charge you are expecting, the MS noise threshold, all of these parameters one need to define depending on the instrument being used and your knowledge about your sample and the runtime. All of these will be crucial factor for deciding these parameters. So, well, then you will go back for further for doing MS search from the same samples, and now you have to look for various parameters, for instance databases, what protein isoelectric point you are expecting, because you would have used off-gel fractionation for example. So, you know which PI value you have used, right, 4 to 7 or 3 to 10. You are looking at what kind of fixed modification, variable modification you may have, what instrument platform one is used, monoisotopic mass, what is the precursor and the product mass tolerance which one need to define. And you have to also mention that how many peptides you minimum want to see, that is it only one or more than one. So, all of these parameters are very critical, and I am sure when you will do analysis today, you will see by changing one by one how much impact that will make in terms of the overall data availability from that. At the end, from doing this, you will generate the protein summary data, and although these things may not be so clear to you to read, but what it generates, that from various spectra which has come from each of these samples, you will see how many peptide sequences you are able to derive. So, those which are in red means are good hits. For instance, the ones in the red are 28, 19 and 18 peptides. It means, of course, as you can understand, these are much more confident hits you will have. You have many peptides for that given protein, and then you are looking at log ratios of I-track labels. So, let us say it was a 4-plex experiment of your control and 3 treatments. So, now you are looking at how 114, let us say that is control, as compared to one of the treatment condition 115, what is the log 4 change ratio available. Then you are looking at 116 by 114, you are looking at 117 by 114 accordingly. So, depending on what reporter ONS you have used, you can look into the 4 changes now. So, all of this information you can generate in the protein summary data. So, this entire analysis is pretty automated. What is important here is your stringency, which is very important, because while you will always think about how good of my coverage is, how many more protein I can get for differentially expressed ones, at the same time what you need to really make sure that your thresholds are very stringent, so that even you have less number of protein, but those should be biologically relevant and they should pass the statistical parameters and the filters. Okay. At the end of doing the entire work, you will derive these kind of sequences, and you will look into the for each one of these peptide sequences, what is the reporter ONS for MSMS, which will help you to do the quantification. So, these are the way of doing data presentation for this kind of I-track workflow. Not every single data you can show in your papers, in your reports, but at least certain peptides you need to show that your quantification looks accurate, your claims what you are telling for the up or down regulation, they are actually making sense from various peptides. So, that you need to show, and you have to report all your data in the supplementary data, and also now you have to upload all your data files into the PRIDE and some other open access databases, where one need to upload all the raw data file what you are generating from the mass spec, because every single data you cannot put in the papers, but community should have access to your data. So, therefore, it is important for you to upload the data in the generic raw data files.